Thank you very much for the organizers to put together this fantastic meeting and also the opportunity to share some of the work. And Anita really made my job easier by introducing many of the concepts that I'll be talking about in my talk. So thanks to that for Anita as well. So our group focuses on trying to understand the dynamics of these two major clinical challenges that cancer patients face: metastasis, which is the spread of cancer cells from one organ to another, and therapy resistance, which, as the name suggests, when cancer cells or cancer patients either do not respond to therapy in the beginning itself or later on come back with tumor relapse and the initial drugs that worked stop working any further. Now, when you think about this dynamics of this process of metastasis, which largely happens through this fantastic set of freeways in our body, the blood vessels, so you can imagine cancer cells in a given organ, we are talking of solid tumors here, they can get onto this freeway and take exits at different organs and try to settle down there and colonize those sites. Now, when you think about what is the efficiency of this process, data suggests that it's actually extremely small, and thankfully so. More than 99.9% of cancer cells which leave the primary tumor are thankfully not able to metastasize, and still it remains the cause of 90% of all cancer deaths. So you can imagine how this number would scale up pretty quickly if this was even a tiny bit more efficient process. Now, why is this process so inefficient? Because the environment around the cells is constantly changing, again going back to Anita's talk, and the cells which have dislodged from the primary tumor are facing a constantly changing biochemical and biophysical environment around them. They don't know where they will end up. They don't know necessarily what exactly lies in front of them, and they are constantly facing changes in oxygen levels and nutrients levels and so on. So what we need to better understand is what is so special about the cells which are eventually able to get through all these bottlenecks and metastasize. So we need a systems level and a dynamical understanding of these processes. Now, as it happens with cancer biology, the first hypothesis every time is mutations. So the cancer cells which are able to metastasize must have new mutations. But clearly that's not really the case, and I'll try to illustrate through this example. So why we have not yet been able to cure metastasis? Now, if you look at just these two pictures, and even if you have not seen the movie, it's actually better if you have not seen the movie. So the first picture is close to the first scene of the first movie, and the second picture is close to the last scene of the last movie. That's where we are when it comes to metastasis. We have the primary tumor, we have the metastatic tumor. We have no clue what happened in between. We don't even know whether people in this scene are the same as people in this scene or not. That is what we know about the dynamics, just these two endpoints, right? So we have very little understanding of when cancer cells leave, why cancer cells leave. Do they leave as individual cells? Do they leave as groups? Is there a size distribution to that? What factors determine those size distributions? Which organs do they stop by? Do they already know where to go, or do they try different organs, right? And how much time it takes for them to have a clinically notable metastasis. These are all largely open questions, which is preventing the development of drugs against this disease, right? And I mean, if you think of major therapies being used in the clinic today for cancer, they go back centuries. Surgery, 1800s. Then if you think of chemotherapy around World War II, radiation therapy around the turn of the 19th century, immunotherapy is the only latest one that is, less than 20 years old. We cannot imagine using a phone less than 5 years old, or certainly 20 years old, but that's where we are when it comes to therapies with this disease that is impacting globally. So for model organisms, we have some answers. So initially, for instance, there was this initial hypothesis that metastasis happens only when primary tumor crosses a critical size, but it has been shown now that that's no longer the case. These can happen in parallel. So there is clinical evidence as well as preclinical evidence from model organisms that one can see circulating tumor cells even before a clinically notable primary tumor is visible. So yeah, so as I was saying, that initial hypothesis in cancer, it begins with mutations, but so far not a single mutational signature has been identified for metastasis, despite spending millions of dollars into this. And there's a very simple reason for this, because mutations are slow and mutations are irreversible. What cancer cells need in this journey is something that is fast and something that is reversible, which is phenotypic plasticity, the ability of cancer cells to keep switching their phenotypes very quickly and very fast in response to the environment that's changing around them. And again, you might have seen many of these images or experimental data where one could segregate these seemingly two subpopulations, but when you culture them independently in the lab, you again get the second subpopulation because there is phenotypic plasticity, because there are multiple attractors in the system and the cells can switch back and forth under the influence of different kinds of biological variation or noise. Now, there's another major challenge which I mentioned earlier, which is, yeah. That's correct. Not really. So people know enough mutations which can predispose the tumor formation, like BRCA1, BRCA2 in breast cancer, but that's only for primary tumor. There's nothing known for what is necessary and sufficient to take one from primary tumor to metastasis in any cancer. So the other scenario is drug resistance or drug relapse. So here is an example. This is melanoma patients. So this is pre-treatment. You can see the lesions in this patient. 15 weeks after treatment, in this case, it's a targeted therapy. So this molecule called BRAF is targeted by this drug called vemurafenib. 15 weeks after treatment, you can see that the lesions seemingly disappear, but only 8 weeks later, in presence of the treatment, the tumor comes back. Right, so we still don't know the dynamics of this very well, and again, Sid has done some beautiful work in this area in colorectal cancer context, and there are these open questions that are there pre-existing resistant cells or are these states enabled by the drug itself, and how can we eventually delay this from happening. And again, this concept of phenotypic plasticity is beginning to get more importance in the field of drug resistance, which again, unfortunately, has been dominated by the genetic paradigm of thinking, the idea that there are some genetic changes which are necessary and sufficient for some cells to show resistance to different drugs. So certainly that plays a role, but that's not the entire picture. There are these non-genetic changes, including some induced by the drug itself, of which I will show you an example later, which can allow for this phenomena to dominate the overall dynamics. So what can happen as a consequence of this phenotypic plasticity, cells switching back and forth, is that you constantly have a moving target. You don't even know the composition of the system at any given time point, and you certainly cannot predict what the composition of the system would be at a later time point because you don't know all the variables, you don't know all the transition rates and so on. And then, of course, this becomes a challenge in the clinic. What do you target that with when you don't know the entire system? And it has been also recognized as one of the hallmarks of cancer in the more recent cancer literature as well. So one can think of phenotypic plasticity from multiple different aspects. One can think of intracellular gene regulatory networks. One can think of population level models to infer transition rates, agent-based models if you want to see how the tumor microenvironment is impacting these. One can think of collecting high-dimensional data and then applying different statistical approaches to infer trajectories and so on. And all of them are being carried out in the field, in the community currently. What we focus on is mostly the first one, and increasingly also on the second one, where we on the last one, where we develop mechanistic models to understand how cancer cells switch back and forth, and use high throughput data to validate some of those models further. We do not want to get stuck to take the forest for the trees. So of course, while we look at each specific cancer in as molecular detail as it needs, we are also interested in figuring out, are there common principles that are seen in phenotypic plasticity networks when you look at multiple different cancers? And also, because cancer is basically development gone wrong, can we also go back to developmental biology examples and see whether some of those patterns are mirrored in those scenarios? So the first example I'll talk about is something called as EMT, or epithelial to mesenchymal transition, where this is thought of as the major engine of metastasis. So if you think of the most common solid tumors, breast cancer, prostate cancer, liver cancer, lung cancer, these are all epithelial organs. They do not have the intrinsic property to start moving and migrating. They're stuck in a specific three-dimensional geometry to execute specific functions. But in order to invade and migrate, they take on some of these properties of what are called as mesenchymal phenotypes or mesenchymal cells in the body and are able to invade the primary tumor. And once these cells reach the secondary organ, they no longer need to migrate, they no longer need to invade, rather they need to settle down and form a secondary tumor. So the hypothesis is that these cells go back to an epithelial phenotype, and this EMT and MET continue to trigger metastasis in multiple different organs. So as a young graduate student trying to find his feet in the literature, when I was going through what is known about EMT in the field of cancer metastasis, all I did was sort of collect all these cartoons that beautifully were represented in these papers on EMT. And these cartoons spanning over a decade tell you an interesting story. There are certain assumptions which underlie all these cartoons. What you notice is that usually it is thought of as a binary process, that cells are either epithelial or mesenchymal. That's one inherent assumption in all these cartoons. The other inherent assumption in these cartoons is that it's reversible, that cells which have undergone a transition from E to M must come back from M to E, because that is essential for colonization to happen. These were the two simple questions we started with. Is EMT really binary? And is EMT really a reversible process? And what kind of data is needed to test out these questions further? So at that point, the kind of data that existed in the literature, largely speaking, and this is just an example, is that people would take epithelial cells in culture, treat them with their favorite growth factor, TGF-beta being the dominant one, go back, come back after 1 week, see the cell shape has changed, and claimed EMT has happened and move ahead. Only 2 time points. And I don't need to tell this audience that if you have only 2 time points, you really can't say what has happened in between. Are there really intermediate states in between, or this was just a monotonic behavior? So what we started doing at that point was put together a minimal gene regulatory network which represents different biochemical interactions that have been reported in the literature for multiple different cancer types, and see what the dynamics of these interactions might look like. So this is, in this particular first instance, you see these 2 transcription factors which are very well known to drive EMT, and these 2 microRNAs which are very well known to drive MET. And the numbers here represent number of binding sites, so you can have some quantitative input-output relationship here. You can convert this into a set of coupled differential equations. This is a slightly different system than just a transcription factor regulation, because you have microRNAs, so you have both transcription and translational processes being affected. So one can think of 1 or more microRNAs being bind reversibly to an mRNA to form these complexes, which can be degraded, or it can be used to sequester the mRNA transiently just for a short while. So you can convert this into a set of equations where there are terms for production, terms for degradation, microRNA regulation, and transcription factor regulation. So what does the model predict? What we predicted was that EMT is not a binary process. And clearly EMT is impacted by many, many other molecules than just these 4 molecules, but even this 4-dimensional simple system or 6-dimensional simple system, if you segregate mRNA and proteins, was capable of giving us something more than a bistable system, in this case a tristable system. So you have this particular state, which is epithelial, which has very low levels of ZEB. You have a mesenchymal state, and also you have this third hybrid state, which at that point, the hypothesis was that, well, this state might enable collective cell migration as compared to individual cell migration, because you have not completely lost your epithelial properties. And then, of course, there are regions of parameters, which is monostable epithelial, monostable mesenchymal, and these are bistable and tristable scenarios. So that was the prediction. And then we started collaborating with experimental colleagues to test this prediction further. So these are lung cancer cells. This particular experimental lab in lung cancer, they had a bunch of 40 different cell lines, and they had measured 2 canonical proteins called ecadherin and vimentin, epithelial and mesenchymal respectively, at a protein level, and showed that out of those 40, only 15 to 20 of them could be classified as purely epithelial or purely mesenchymal. The rest of them had both kinds of proteins at a bulk level. So we wanted them to look more carefully whether these 2 proteins were co-expressed at a single cell level. So in this particular example that I'm showing here, red and green respectively, ecadherin and vimentin have been mapped, and you can see this co-expression continues over multiple passages in culture. It's not just a snapshot that is seen here. You can quantify this further. You can change the cell line. You can look at breast cancer, prostate cancer, lung cancer, colorectal cancer, and quantify the percentage of cells in each of these quadrants, which are pure epithelial, pure mesenchymal, or the ones which are double positive. And interestingly, when these subpopulations are segregated and cultured independently in the lab, you see that they switch because there is spontaneous switching that is happening, which is again the outcome of having this multi-stable dynamics that you could see this switching. And this has been seen in many, many cases now. Sure.
I guess I'm just trying to like understand this in the sense that, I mean, this network presumably has a bunch of other components as well, right? Like it's like, as Anita showed, like it's really a complex network. And the prediction that it's kind of, multi-stable is coming from this. I mean, how generic is that statement in the sense that if you include many other components, or, many of these parameters are presumably like, have to be chosen, how sensitive is it to all of these different choices?
Just give me 3 slides.
Okay,
I'll come to that question. Beautiful question. Yes, in some, at least the mesenchymal cells secrete TGF-beta, which can then convince neighboring cells to. Yeah, so there is some evidence of frequency-dependent switching rates as well that we have seen in the latest analysis. So yes, coming back to the exact question, now there are many, many other models, many other networks that have been made. That was just a 4-node network. People have made all the way to 18-node networks. Some have done discrete Boolean models, some have done ODE models, all different kinds of strategies in the past decade. And consistently you see that there is one or more hybrid states being reported. So it's not a binary process. That is something which has been reasonably well established in the literature. This work that I'll talk about later as well, using Ising model, also you can, when you project all the solutions on a principal component, you can again see that there are these cells which are epithelial and mesenchymal, but then it's a large number of states which are somewhere in between. So the next question we started asking was, well, does it matter? Okay, these states exist, so what? And we did a similar approach of trying to couple what was known in the context of stemness, so the ability of cancer cells to start a new tumor. And our model predicted that it is these hybrid cells which actually have the maximum stemness as compared to purely epithelial or purely mesenchymal. And this was just a prediction until it got tested a couple of years later in breast cancer and in skin cancer, showing that, of course, there are multiple states of EMT, but the ones which are most metastatic are not the ones on the either extreme, but the ones which are somewhere in between, which is exactly what our model had predicted. And more recently, there was a beautiful experiment done which actually showed that it is the plasticity which matters more than heterogeneity. So they were able to generate cells which are locked epithelial or locked mesenchymal by doing CRISPR and various other perturbations in the system. And when you do locked epithelial or locked mesenchymal, you don't see tumor formation, even when you mix epithelial and mesenchymal, which are locked. So now the system is heterogeneous but not plastic. You again don't see tumor formation. But the moment you have these plastic cells, and you suddenly see a spike. So there's very neat distinction to be made here, because often, at least in the experimental literature, these terms are used interchangeably, but they are not, because plasticity can give rise to heterogeneity, but heterogeneity may not arise only due to plasticity. There can be various other factors to that. There's clinical relevance. There is more and more literature that single cell migration is actually very rare, if at all, in any cancer. So people have done 3-dimensional reconstructions of multiple tumor biopsies and shown that what were presumably thought of as single cells were actually just projections of branches coming out of that. And also clinical evidence that only 2% or 5% of hybrid cells can actually drop the survival dramatically in patients. So hybrid cells need not even dominate the landscape. As long as they're present in a small spectrum, you could actually see this clinical association. Of course, based on clinical factors, you can't say anything related to the causative evidence, but nonetheless, there is evidence for that. So in the field of EMT now, which was initially thought of as a binary process, now what we know over the past decade or so is that it is not binary. Actually, there are multiple states in between, and these multiple states in between are actually the most metastatic ones. And also there was a recent study which actually quantified the plasticity as well and showed that the ones are in between are actually the most plastic as well as compared to the ones which are completely epithelial or mesenchymal. Please.
How do they do these? Could you briefly describe how do they actually do
These kinds of experiments, right? So these kinds of experiments are usually done by multiple surface markers. So because if you use only one surface marker, you can distinguish two phenotypes. So if you do multiple surface markers, you can have maximum of 2 to the power n different distinctions. And then you segregate these subpopulations and do in vitro or in vivo injections, and then notice again, yet again, do the flow cytometry or quantification of this and map the dynamics of that. So it's actually a very similar sort of extension of this experiment, where this was using only 2 markers. So you start from only one of them and then see what is the fraction of that population as a function of time. But if you can have many more, then you can expand this high dimensional space.
And question back here. So plasticity, you've shown the different basins, but the mechanism of switching, is it stochastic, or is it because there are kicks that are deterministic that push it between the different basins?
It's a mix of both. So like for instance, hypoxia can trigger EMT to happen. Also TGF-beta, which can also be secreted by cells which have undergone a switch. So there's an autocrine or paracrine feedback loops also acting. Matrix stiffness can induce EMT. So there's a whole bunch of different factors which can induce this. So then we were intrigued by this observation that these hybrid cells are most plastics, and we wanted to look more into it. Can this be explained by gene regulatory networks? I mean, of course, you can give a sort of a hand-waving kind of explanation that because they're in between, it's easiest to switch. They can go to epithelial or mesenchymal, but is there something deeper than that is what we wanted to look at. So again, going back to the question that was asked about, how generic or universal are these properties if you look at many, many networks. So we did look at many, many networks, starting from 4 node, we have looked at all the way to 57 node networks. And each of these networks was put together by a different group in the community. So we didn't really put together these networks, but we wanted to ask this question, that is this high plasticity behavior a direct outcome of the topology itself? Can we just look at the topology and again 4 different networks and explain that this happens? So because to cut down on the computational cost, we moved ahead with a simple Boolean model and a simple Ising network, Ising update, where because we don't know the exact update rules for each of these nodes, we said that each node can be just +1 or -1, and you can update each node if the number of inhibitions is more than the number of activations and so on. Please. Computationally or experimentally? Computationally, I'll explain that. Just give me a minute. Yeah. Right. You can identify different kinds of cells. Yes. And is it the numbers of cells that tell you how deep each well is? Is that the? I wish it were as quantitative, but I think this is just a schematic representation of that biological data. So we are actually trying to get more quantitative details and trying to see whether the sort of frequency of states frequency of initial conditions that converge to a state, can that be used as a metric of plasticity? And so. No. One can take this idea seriously that these 2 are deeper than the other 3 in between. The extremes are deeper than the ones in between. That's the only thing that one can take with some quantitative detail. Other than that, what varies among these 3, I don't think we can take with as seriousness. So we looked at this network, and before doing any simulations, we wanted to see, is there any structure in the network itself? So we took one of these networks, and we have done this for other networks. I'm just showing this for example. And we represent this in the form of a matrix, where each row is the source and each column is the target. So you have this 22 cross 22 matrix in which 82 elements are filled, because there are only 82 edges, which are either +1s or -1s, and the remaining ones are zeros. So that's this matrix. And as we very well know that in biological networks, again going back to Anita's talk, you don't really need to be in direct contact with a node to be impacted by that node. You can have the influence propagate through multiple nodes in between. So we took this matrix and we multiplied this matrix to itself many, many times. And basically, now this is a matrix polynomial. And let's see what happens. And you see a very clear structure that emerges. The structure is the following, that each epithelial node is effectively activating each epithelial node. Each mesenchymal node is effectively activating every other mesenchymal node. Every epithelial node is effectively inhibiting every mesenchymal node, and every mesenchymal node is inhibiting every epithelial node. So at the end of the day, this is a very simple structure, that there are these teams of players which are trying to pull the system to being either epithelial or mesenchymal, and the poor hybrid cells don't have any teams, so that's why they get pulled on either extremes. So we wanted to quantify this further and said, let's first check whether this behavior of teams is this unique to biological networks. So we took this adjacency matrix and generated many null models of it by swapping the values of +1s, -1s, and 0s and generating non-biological hypothetical network. And then again generate influence matrix equivalent of that by applying the same formula. And we clearly see that the biological network shown with red here lies much ahead of most random networks. So it's the phenomena of teams is actually specific to biological networks and cannot be seen in any just random network, at least in this example. So now we did the simulations further, and going back to the question, how did we quantify plasticity? We defined frustration. Frustration is basically, again, directly taken from the spin glass systems. We define as frustration of a state as how much does it mismatch, how much does a state mismatch the rules that has been given. So again, it's on a scale of 0 to 1. So we observed that there are these phenotypes which have very less frustration, and there's another set of phenotypes which have very high frustration. And these phenotypes, because we know the molecular identity of each state, you can also classify them as epithelial, mesenchymal, or hybrid. Hybrid states are more frustrated, less frequent, and epithelial states and mesenchymal states are less frustrated and more frequent. Then you can ask this question where you can start from an epithelial state, mesenchymal state, or these hybrid states and flip n number of nodes randomly. You can ask, on average, how many nodes would you need to flip so that you leave a state? If you start from epithelial and mesenchymal, that is around 40 to 45%. If you start from hybrid, that's 5%. So the hybrid states are much more plastic to begin with because there is no reinforcement, there is no resistance offered, which is offered in the presence of the teams, because there are many more players trying to dominate the same attractor and trying to pull back. So the restoring force is much less in the context of hybrid states because there is no teams structure that is enabled there. So now we have been thinking about these questions. Well, why do teams exist? So one is, of course, to think about the rates of cell state transition. The other question, going back to what I started with, cells need to coordinate their behavior. They need to change metabolism, EMT, stemness, all of these properties together so that they can overcome all these different bottlenecks. And that can be one other instance where the idea of teams can be really relevant, that when cells are undergoing EMT, they are already carrying some other properties with them which might be helpful for them in order to complete the metastatic cascade. And I'll just show you a couple of examples of the same. One example is this of drug resistance to tamoxifen. Tamoxifen is the first targeted therapy that came into the market in 1980s. What tamoxifen does is it binds to estrogen receptor, and it prevents estrogen receptor binding to estrogen. So this is used for treatment of ER positive or estrogen receptor positive breast cancer, which is the most common subtype of breast cancer. And when you look at, so this estrogen receptor has two different isoforms. They regulate each other in different ways, and also many players connecting EMT. So we put together a similar network, do simulations of this where when we write down a set of coupled ODEs, we randomize the parameters to try to look at randomized parameters and initial conditions to see what is the possibility space of phenotypes. And what we notice here is that if you quantify an EMT score, which is ZEB minus MER200, and you quantify a resistance score, which is ER66 minus 36, again, this is based on clinical data as well as preclinical evidence, you see there is a strong correlation that when cells are epithelial, they are sensitive to the drug, and when cells are mesenchymal, they are resistant to the drug. But interestingly, these processes can also drive each other. So because of these coupled feedback loops, when you perturb one node, you are also perturbing the system in other ways. And then we looked at publicly available data transcriptomic data where EMT was induced in these breast cancer cells using different modalities in different studies. In all of them, estrogen response was shut down. And vice versa, when you shut down estrogen receptor, EMT goes up. Or in this other study where resistant sublines were generated by constantly exposing the drug to slowly increasing concentration of the drug, you again notice that EMT goes up. So these ideas that you have these feedback loops, and these feedback loops are actually coordinating cellular behavior. So think of the clinical implications of this. Patients who are receiving this drug, a subpopulation of those cells are actually becoming more metastatic because of the drug. If the drug was not given, probably this transition would not have happened. We don't know the details of the following as to what is the frequency of cells which undergo this transition, at what concentration of the drug, and what prior treatment that was given. So those are things which we are still working on. But clearly, this is an example where you can see that seemingly different properties of cancer cells are actually strongly coordinated in phenotypic high-dimensional space because of the existence of these kinds of structures. You can expand this to more than 2 behaviors. You can look at EMT, tamoxifen resistance, and stemness. And again, you can continue to see these teams behavior, and this is unique to biological networks and not really seen in random networks. This is another example, EMT, tamoxifen resistance, and PD-L1, which is a target for immunotherapy. And again, you can notice that when cells, when you silence estrogen receptor, EMT goes up, PD-L1 also goes up, which means that the efficiency of immunotherapy also goes down. So there is this cross-resistance, and there is a reason for this cross-resistance because these molecules are impacting each other. It's not happening randomly anywhere. And that is the idea we are trying to drive here, that you can understand mechanistic underpinnings of these cross-resistance and hopefully use these frameworks to get to a more efficient way of identifying synthetic lethality combinations by looking at what exactly the system is responding to. So what we know now is that these hybrid cells are the fittest for metastasis because of all these different properties, and some open questions which we are still continuing to working on is how many states exist, what are their molecular footprints on other aspects, and how can we target them clinically. The second question was, is EMT a reversible process? And there was again some experimental evidence that if you induce EMT for say 5 days and you withdraw the signals, the cells come back. But if you induce it for 8 days and you withdraw the signals, the cells do not come back. So there was some tipping point kind of behavior reported in the literature, at least at this bulk level, showing that there might be some transitions that are happening here. So we made a very simple model where we took this Hill's coefficient in these regulations, which is usually, not Hill's coefficient, Hill's threshold in the denominator, which is usually a constant variable, and we made the constant parameter, and we made the dynamical variable. And the idea was that many of these transcription factors are known to be so-called pioneer transcription factors. They can really remodel the chromatin in such a way that even when their expression goes down, they can continue, because the chromatin has been changed, their impact can live much longer than that. And using this very simple model, which was based on a previous model by Kunihiko Kaniko's group in the context of reprogramming, we were able to show that you can change the population distributions by just impacting this particular link where we implement this kind of epigenetic feedback. So you could actually have EMT and MET, both can be irreversible because both of these transcription factors and other nodes which we included later in the network can show these kinds of behaviors. So then we started thinking more carefully about these experiments and asking these questions, that is EMT induction always irreversible? And this is where our collaborator Mike Tonoff did these experiments where you induce EMT for short durations and withdraw the signal. But when you induce EMT for longer durations, you wait much longer. And we saw a beautiful trend here that, so this is time after withdrawal, and this is the expression of E-cadherin, which is in some sense a percentage of cells which actually come back. So if you start from the short induction, of course the cells come back pretty quickly. It's a linear response. Interestingly, in the long-term scenario, you see a delay. If you would have stopped the experiment somewhere around 20 to 25 days, you would have claimed that this is an irreversible reaction. But here, the cells actually start coming back only after this delay, and then you notice that cells do come back. So the rate of switching back and forth clearly depends on what extent of time it was initially induced. And these things need to be looked at much more carefully rather than just saying a process is reversible or irreversible. And again, we were able to explain this using where we coupled this bifurcation diagram that I showed you earlier together with cell division. The idea was that every time a cell divides, there is some loss of memory that happens. And because these experiments are on time scale of many, many days when cell division is happening, that could move you back and forth in this bifurcation diagram, could also change the structure of these bifurcation diagrams, so you can actually see the population structure completely changing here as well. So there is a delay in return of population, but that delay is finite in nature because with cell division, you're constantly losing memory as well. And there has been some recent experimental data where EMT was induced for different durations and then the signal was withdrawn. And when you take the ATAC-seq data of that, which is chromatin accessibility profiles at a high dimensional level, and you project them on a PCA, you notice that the cells which were induced for short durations, which is seen in blue here, they actually come back to where they started. But when you look at the long-term cases, they actually segregated. So there's also hysteresis that one can notice even in this high dimensional data. So hysteresis in EMT has been experimentally shown now at transcriptomic level, at proteomic level, and also recently at a chromatin level. So all these ideas about multistability are beginning to very clearly show evidence in the experimental data as well. So what we learned here is that cells can be resistant to undergo EMT or MET. Some cells might just not respond to EMT. And going back to the question that Mukund was asking, how deterministic or stochastic that is, that depends on the previous observations that they have had. And also there can be cell-cell communication factors, which I'm not talking about today, but we have some data showing that these rates of transitions can be frequency dependent as well in the population. Now, we live in a data deluge era. There is huge high-dimensional data being collected. Sometimes I wonder which time is better. Is this time when we have a lot of data and we don't know what to do with it, or previous times when we didn't have any data and we were just making models to hopefully collect more data in this case. So now there is more and more data coming out in the EMT field that there can be not just one trajectory, there can be more than one trajectory during EMT. There can be hysteresis, and that hysteresis can be seen at different levels of regulation. There can be different rates of EMT depending on intrinsic states and so on. And we have also collaborated with some of these groups listed here in trying to make a better sense of this high dimensional data. But what we are now currently looking at is that can we do this in a more meaningful manner rather than just churning out irreproducible UMAP and t-SNE plots, which get published in all the top tier journals these days. But the moment you run it next time, you may not see the same pattern because it's stochastic by nature. And so can there be reasons behind this? And one thing we are actively working on is that perhaps teams, the idea of teams of nodes or genes can be a meaningful dimension reduction method which could help us see all these patterns in this high dimensional data. And this beautiful article which came out last year in the context of cell-state transition in many different contexts, not just EMT, makes this call that it's time to Keplerize the cell and to discover these general patterns that can better understand what exactly is going on rather than just keep going on collecting more and more data. So when going back to this network with teams, when we simulated the dynamics of this network using ODEs over multiple parameters and multiple initial conditions, and then we projected them on a principal component, each dot is an enumeration of a given initial condition and given parameter set. You notice that PC1 explains 95% of the data. And when you do this for all the random networks, this explanation comes somewhere around 30 to 40%. And again, in this plot, x-axis is team strength, which can be calculated without doing any simulations. And the y-axis is PC1 variance, which can be done after doing the simulations. And each dot here is a random network, and this is where the wild-type network lies. So you can clearly see that the stronger the team strength, the higher the PC1 variance. There's a clear structure to this. And when you look at what exactly is PC1 composed of, so PC1 which is a linear combination of many of these variables, you can notice that all the mesenchymal variables have a negative coefficient, all the epithelial variables have a positive coefficient. So even the team's information is present in the PC1 eigenvector directly itself. But again, this is not seen in any random network. You can look at real transcriptomic data as well, which we have done, and that can also be explained largely by PC1. And then we have done these kinds of sort of in silico experiments where we replace each relevant gene with a housekeeping gene, which is not connected to EMT, and the PC1 variance continues to fall down dramatically. This is not specific to EMT. You can take other examples, gonadal sulfate determination, small cell lung case network, pluripotency network, all of them we see the same behavior, all of them biological network lies far away from the random network. So this might be a more generic principle rather than something which is just reported in the context of EMT as well. We're currently also looking at artificial networks. So let's say you have a network of which you can decide that it has a team structure. You can fill it in with different densities and ask the question, what is the relationship of network density with PC1 variance or network density with team strength? And there's a beautiful trend that shows up that all a network needs is somewhere around 30% density in order to achieve a team strength of 0.5 or more. And again, a similar sigmoidal behavior is also seen when it comes to PC1 variance. We are still thinking about what happens when you don't have a uniform density, but you can allow different densities in each of these different squares that is mentioned here. Please. So two questions. So one is the hybrid, how do hybrid cells evade immune response? So there are multiple mechanisms. One that we have looked at is by upregulation of this molecule called programmed death ligand 1 or PDL1, which is a cell surface receptor. And so if you look at the immunotherapies, there's a like anti-PD1 or anti-CTLA4 therapy. So what they do is that they sort of unleash the T cells to unleash the immune cells to attack the tumor cells. But if the PDL1 sort of acts as a brake, so if PDL1 levels are higher, then PDL1 interaction would prevent that cell death from happening. So that's one mechanism. There are other mechanisms also that people have studied. The idea of teams was present in the network structure itself, that even if you don't do any simulations, you just look at the adjacency matrix and multiply it many times to get the influence matrix, and then do the typical hierarchical clustering. Do you see a structure? And the answer in that case was that yes, we do see a structure, and that structure was such that every member within a team was supporting other members, and every member across teams was inhibiting other members. So that's the general toggle switch. So it's a high dimensional toggle switch at the end of the day in that scenario. So we're currently thinking of teams as a way which can minimize frustration in EMT and many other networks, and that is why we see only a limited number of trajectories and only a limited number of cell states, despite a huge combinatorial complexity that might occur. And that, of course, reminds us of the protein folding question, that despite so many possibilities, why only few of them are seen here, because there is constant canalization and constant elimination of many other possibilities because of the cooperativity that's built into the network, and teams is a way to build cooperativity in gene regulatory networks. The concept of teams need not be restricted to what's going on within a cell. You can think of teams of cell types in a tissue. So in this particular example that we had looked at earlier, when we didn't think of the concept of teams, there are epithelial cells, mesenchymal cells, and these M1 and M2 macrophages. Each of them is secreting different cytokines to impact each other's behavior. So the formulation is slightly different, but nonetheless, we still see this bistable system where epithelial cancer cells usually exist with M1 macrophages, and mesenchymal cancer cells usually exist with M2 macrophages, for which we had done some initial analysis in the Cancer Genome Atlas data at that point of time. And something we are working on right now is, even in the field of macrophages, we now know that M1 and M2 are just two extremes. There can be many more states in between. Can these states be one other way in which these hybrid states might be stabilized in a tumor as well? So I'll just summarize that in different instances that we have looked at, we do see that there exist design principles, that there is multi-stability, which could explain the experimental observations of phenotypic plasticity and heterogeneity, and the existence of teams could allow for higher or lower plasticity, and could also allow for cell behavior to be coordinated rather than just being some random combination of different possibilities in this case. With that, let me thank our team of students and collaborators across these different institutes, across different disciplinary backgrounds as well, with whom it's been a privilege to work and try to decode some of these answers. And thank you once again for your attention. I'll be happy to take questions.
Clearly. So should I be thinking of it as, so let's get back to
Let me just pull up that slide. Yeah.
So should I be thinking of this as you're basically saying this is your adjacency matrix, and then you go a to the power n, let's say a is the adjacency matrix to the power n, to see basically what happens if you just pop a gene through a network again and again and again, so that like, and then you have some sort of like a dynamic here. Is that correct?
So it's actually a polynomial. So it's, adjacency matrix plus adjacency matrix square plus cube plus all the way till that. And then we penalize it by the length of the path. So the longer a path is, because of course the influence keeps getting down, we just have it in the denominator.
Okay, that makes sense. Second question was about how you were doing some of the work with the networks, where you're saying, okay, this is my network, and here's like, some null networks which I'm generating. What sort of care do we have to take in setting the statistics of these random networks to make sure that we're not like, either over encoding or under encoding information into our, like, into what we want to know? So,
right.
Like, can you talk a bit about that?
So I can tell you the conditions that we have taken. So of course, this is the same size, same density. We also control for the same in degree and the same out degree of each node. But what we do not control for, and that's what gives this variability, is that how many of the in degree is inhibition versus activation, or activation versus. So the total number of activations and inhibitions are the same because it's the same number of reds and blues and whites. And the in degree is also the same, but the distribution of in degree in terms of how many activations versus inhibitions for each node is not controlled for. And that's what gives you this variation.
Thank you.
I may have missed this right at the beginning. These networks are the same in healthy cells or these tumor cells, right? So what prevents this happening in healthy condition?
So these networks so far have been seen mostly in tumor cells. The healthy cells equivalent of this has not really been built in as detailed comparative manner as we would want to be. The underlying idea here is that in diseased cells, there is much higher plasticity as compared to the normal cells. And we have some unpublished data for that, where we have this comparison of team strength in metastatic versus non-metastatic conditions. And we see that the metastatic patients had much weaker team strength, which could explain the higher plasticity.
That goes back to your original question. Then there's some previous determinant, right? So, and that should show up as either a mutation or yet another sort of plastic variable. So what could that be that distinguishes these cells from the healthy ones?
Right. So the mutations can certainly make cells more plastic. That's at best, is what our current hypothesis.
Very quick question. So why would you think these are attractors and not ghosts?
Attractors and not?
Ghosts.
Good questions. We'll have to think more carefully about that.
So Mohit, maybe I'll ask a quick question following up on Mukund's question, right? So, I mean, in a completely different context, because we are interested in circadian clocks, right? If you just look at the set of genes that are known to generate the clock, we see exactly the same kind of team's behavior. This is in healthy, nothing to do with canalization, right? No cell states, anything, right? So we see exactly that kind of structure in gene expression matrices.
So in gene expression matrices, yes. So that's why I think just looking at gene expression matrix might not tell you the exact picture, because that can come due to other dynamical outcomes as well. But if you compare gene expression and network topology simultaneously, and you continue to see this behavior, then one can claim that this is coming because of the dynamics of the network topology.
And what do you mean by take them simultaneously? I didn't
So
understand.
in the sense that if you look at, so in the networks that you're looking at, do you also see co-expression patterns in the network structure itself?
I mean, we
In
have
the cell circadian rhythm networks.
I mean, you have direct connections
Right.
between one gene and another. Is
Right.
that what you mean by?
And then if you generate this sort of influence matrix.
This we've not generated.
Okay. So then
okay.
That's what I'm saying. Looking at influence matrix generated from networks and looking at transcriptomic data
So if you
from the same context.
So if I understand you right, then you're saying you could see these kinds of teams just in the gene expression space,
which may not come
which may not come up
yes.
here. I see. This we've not checked.
Yes.
Okay.
Yes. Yes. So teams is not the only way in which you can get that co-expression pattern. I mean, the whole statistical approaches of weighted co-expression gene like WGCNA and all, that's not necessarily built on the teams concept. That's built on co-expressed modules which are changing in different ways. So we're trying to find where the statistical approaches and dynamical approaches can sort of intersect and tell us more about why we see what patterns we see kind of questions. But yeah, we have seen, again, going back to both, I think what Mukund and Chan was saying, we have seen teams in developmental examples as well. So there's nothing unique to this about cancer.
Yeah, I just have a quick question. So I am not understanding correctly. This team thing is only obtained from a snapshot data?
So teams is obtained without doing any simulations of the network. But the network was, of course, built on a bunch of experimental data where each node was perturbed, and then you look at dynamical data as well.
Okay, so you are thinking that this team is a property which won't change with time? The adjacency
so that's a good point. Yeah, so we have
over the time it's sort of robust, or you think that it is going to change with the time?
So the current assumption is that an activation will not convert to an inhibition, but the strength of activation can change.
Okay.
So the team strength can go up and down, but there are no cheaters. Okay. But we have also looked at scenarios where you introduce cheaters, and then the team strength actually begins to fall. There's more penalty to being a sort of a promiscuous player than being absent in the network structure.
Maybe we have to figure out something else to quantify that. And all. Thank you so much.
So just curious, these, when you multiply these matrices many times, basically you're enhancing the largest eigenvalue.
Yes, yes.
And have you looked at the largest the eigenvector corresponding to the largest eigenvalue of these and other eigenvectors?
Exactly. So that's this example. So this is the principal component eigenvalue, and this has the same information as that of teams. Mm-hmm. This even tells us more information in the sense that who is contributing more to the team, right? Which cannot be gauged just by looking at the influence.
That's right. Okay, fine. So this is exactly that.
Yeah, exactly that. Yeah. Fine. We're doing a spectral analysis in artificial networks also to establish this concept more generally. Sure. Sorry?
Point.
So, I mean, so we have done some analysis of sort of early warning signals and tipping points, looking at autocorrelation when you add noise into some of these same equations that I showed earlier. And we do see that those early warning signals do show up associated with each of these tipping points. Yes. Yes. Right. Right. So we have looked at slightly different networks as well, or even when we change some parameters in this equation, what happens is that you basically take these two arms of the bifurcation diagram and you pull them. So then you lose tristability, but what you have is bistability, monostable hybrid, then hybrid and mesenchymal, and then monostable mesenchymal. So depending on the structure you're looking at, you can get different combinations in this case. And again, we have done tripping point analysis in that, and also early warning signals show up. These are all saddle points in this bifurcation.
I would say let's break for lunch, and then maybe we'll be back at around 2:27 or so for the afternoon session.